Okay, perfect. Yeah, hi everyone. As Professor Markus said, I'm Natalia. I'm a PhD student in the stroke research group here at Cambridge, and I focus on identifying markers to predict disease severity and progression in CADASIL. So I wanted to briefly introduce myself. Just one moment. Screen's in the way here. Okay, here we go. Briefly introduce myself. I did so at the most recent in-person meeting, but I wanted to give another quick introduction for anyone who I haven't met yet. So I'm originally from Seattle, Washington in the United States, where I studied neuroscience and started getting involved in research. Specifically, my first research experiences were actually on how the brain processes visual information. But then I moved to Cambridge to pursue a master's in clinical neuroscience and worked on Huntington's disease, which I'm sure many of you are familiar with. It's a hereditary neurological condition. It's primarily movement disorder. And for my master's project, I worked on identifying molecules in the blood that were able to predict how severe the disease would be for a patient and how quickly the disease would likely progress for them. At the same time, I became really interested in stroke research. And so last year, I started my PhD in the stroke research group here at Cambridge. And as I mentioned, my projects focus on CADASIL, which is what I'll be talking about today. And I also just want to take a moment again to give a huge thank you to the CADASIL Support UK leaders, Sophie, Karen, and Glenn, who supported my application to a Stroke Association PhD scholarship, which, as Professor Mark said, I'm very pleased to share that I was awarded. So this will help fund my PhD and provide funding for one of the main research projects of my PhD, which I'll explain in a bit. But I just want to extend a huge thank you for their support. So first, I wanted to give a little background about CADASIL, though I'm sure most, if not all of you are already quite familiar with this, but it does help to explain the motivation behind some of the questions that I'm trying to answer with my research. So CADASIL is a genetic disease that affects the small blood vessels in the brain that are responsible for delivering oxygen and other nutrients through the blood to brain tissue. And it's caused by a mutation in a gene called Notch3. And in CADASIL, there are two main characteristic features that show up on MRI scans, which are lacunar infarcts, which are these small holes in the brain. Hopefully you can see my mouse indicated by these yellow arrows that are a result of that lack of blood flow to these areas of brain tissue. And the second is white matter hyperintensities, which are essentially a scarring of brain tissue. And these are these brighter regions here. And in terms of symptoms, migraine, strokes, and cognitive impairment can all occur, but importantly, the symptoms of CADASIL can vary a lot from person to person, even among people who have the same genetic mutation. So for example, you could have one person who has a stroke at 40 and develops dementia at 70. Meanwhile, someone else with the same mutation might not have a stroke until their 60s and may never develop dementia at all. Furthermore, mutations in the Notch3 gene that are typically linked to CADASIL are actually much more common in the general population than previously thought. And those in the general population who have these types of mutations do show an association with the occurrence of stroke and dementia. However, they have much milder manifestations of the disease. So this raises an important question. Why is there such wide variability among people with these CADASIL-associated mutations? And what can this tell us about what drives the severity and progression of the disease? So this also leads to some important practical questions, like how can clinicians offer more personalized care and give patients guidance on family and life planning, given this wide variability? And this also affects how we develop treatments. So in addition to selecting the right targets for treatment, clinical trials have to select patients who are at a higher risk of developing symptoms sooner. This way, researchers can test whether treatment works within a realistic time frame and without putting too much burden on the patients, since things like MRI scans and spinal fluid tests can be quite invasive and time-consuming. So there's a clear need for a more convenient, accurate way of assessing someone's risk. And one promising approach is to look at blood-based biomarkers, specifically the levels of proteins and metabolites in the blood at different stages of CADASIL. So far, only a few studies have looked at these markers in a really rigorous way, but the early findings are encouraging. they suggest that these blood markers could be really useful for predicting how severe the disease might become and how quickly it could progress. So I just wanted to give a very simple explanation of why looking at proteins and metabolites can be helpful. So you have your DNA here, which stores your genetic information, and your body makes a copy of that information into something called RNA. And RNA is then used as a template to make proteins. And proteins are the molecules within our body responsible for almost every biological process and function of the body. So as they do their jobs, they go through various changes and chemical reactions. And this activity produces smaller molecules called metabolites. And metabolites act as essentially either the fuel for these processes or the end products that result from them. And they include things like sugars, amino acids, and fats. And because proteins and metabolites reflect what's actively happening in the body, they can provide really valuable insight into what happens when there are abnormalities at the genetic level, like a mutation in the Notch3 gene. So what exactly happens on a genetic level in CADASIL? Well, we know that CADASIL affects the Notch3 gene, which contains the instructions the body uses to then make the Notch3 protein. And this protein, which is illustrated right here, is a receptor protein. So it's really important in signaling and is made up of a large extracellular domain and a smaller intracellular domain. So one part that exists outside the cell and another part that sits inside the cell. And it plays a key role in the function and survival of vascular smooth muscle cells, which are muscle cells that surround blood vessels. As you can see here, they're the cells in purple. And they sit outside of this thinner layer of endothelial cells, indicated here in sort of this pale yellow beige color, that serve as the inner lining of the blood vessel. So there are different types of mutation variants within the Notch3 gene that can lead to CADASIL. So specifically, there are different mutation variants that affect different areas of the protein's extracellular domain. However, what almost all of them have in common is that they change the number of these specific building blocks called cysteines within the Notch3 protein. And so cysteines are indicated here by these small green circles. So when there are too many or too few of these cysteines, this disrupts the structural integrity of the protein, causing it to misfold and reducing its ability to bind properly to other molecules, which then causes problems in signaling between cells. This also leads to an abnormal buildup of this extracellular portion of the Notch3 protein at the surface of these vascular smooth muscle cells. And over time, this leads to degeneration of these muscle cells and progressive small vessel damage, though the precise mechanisms behind this are still not fully understood. And though we don't know exactly how biological processes or pathways are affected and to what extent, a lot of research has shown that one of the most consistently affected pathways in CADASIL involves something called the extracellular matrix, and so also known as the ECM. So the ECM is basically the support structure that holds our cells together. So more specifically, it's a network of proteins and other molecules that surround cells and give tissues their structure and help them function properly. So you can see here we have our vascular smooth muscle cells, which are surrounded by various proteins like collagen, which is indicated in purple here, and elastin, which are these yellow structures that help maintain the strength and flexibility of the vessel wall and partially make up the ECM. And the endothelial cells that we have here, which is that inner lining of the blood vessel, are also supported by the ECM via this layer called the basement membrane. And so in CADASIL, research has shown that the extracellular matrix becomes disrupted, meaning that the structure and balance of the support system starts to break down. And this can contribute to the vessel damage and reduced blood flow we see in CADASIL. So we're still studying exactly how the mutations in the Notch3 gene lead to these ECM problems, but one theory is that the abnormal buildup of the misfolded extracellular domains of the Notch3 protein that I mentioned earlier at the surface of these vascular smooth muscle cells may trigger inflammation and scarring in the ECM, which then weakens the vessel structure over time. And this breakdown of the extracellular matrix doesn't just weaken blood vessel walls, it also affects another critical structure, which is called the blood-brain barrier, which you may have heard of. It's essentially a protective barrier that controls what can pass from the blood into the brain, and it helps to keep harmful substances out. So it's made up of tightly connected cells, including endothelial cells and the basement membrane of the extracellular matrix. And in CADASIL, when the ECM and basement membrane become disrupted, the support for these tight junctions here weakens. And as a result, the blood-brain barrier can become more permeable or leaky. And this means that substances that normally wouldn't enter the brain, like inflammatory molecules or toxins, could slip through. And this leakiness can contribute to further inflammation, further damage to blood vessels, and potential worsening of neurological symptoms. So understanding how both the ECM and the blood-brain barrier are affected in CADASIL is really important because it not only helps us explain the disease's symptoms, but also points us towards new treatment strategies that aim to protect or repair these critical systems. So one of my main PhD projects is aimed at addressing these questions about exactly how these and other pathways are affected in CADASIL. And I'll do this by analyzing how proteins and metabolites behave in people who carry Notch3 mutations typically associated with CADASIL. So, I mean those cysteine-changing Notch3 mutations. So to do this, I'll be studying two different groups of people. The first group includes patients who have taken part in our familial small vessel disease study in Cambridge. And the second group comes from the UK Biobank, which you may have heard of. It's a large research study that includes data from around 500,000 people across the UK who have shared a wide range of health and lifestyle information. So it's a really useful resource to look at. And for both groups, I'm comparing blood marker levels with several other types of data, namely medical history, and this is both of the individual and their families. So I'll be looking at whether they've had migraines, strokes, or dementia, and when. And I'll be looking at genetic information, so what type of Notch3 mutation they carry, imaging data, and this will mainly be MRI scans. So I'll be looking at different imaging markers like those lacunar infarcts and white matter hyperintensities that I mentioned earlier. And lastly, I'll be looking at cognitive function, so how well their memory and processing skills are working and how these are all related to their blood profile or their levels of proteins and metabolites. So during the first year of my PhD, I focused on analyzing the data from the UK Biobank. So now I'll share some of the most interesting findings from that part of the project. So I'll start off with the protein levels. There were 147 proteins that showed significantly different levels between people who carry the genetic variant and those who don't, so the control group. So all the small circles here that you see on this plot represent the levels of a single protein. And the colored circles represent proteins that were found to have significantly different levels in variant carriers compared to controls. And I just want to point out that there's this center line here right at 0. And if a circle is on the left side of the central line, it means that the protein level was lower in variant carriers. And if it's on the right side, then that means the protein level was higher. So as you can see, most of the significantly different proteins had lower levels in variant carriers compared to controls. And we found that 11 of these proteins were associated with family histories of stroke and dementia, and 2 of them were linked to white matter hyperintensities, which again is that scarring of brain tissue that I mentioned earlier. And these 11 proteins are mainly involved in inflammatory and angiogenesis pathways, which in other words are pathways that help regulate the immune system, which is a vital part of how the body repairs itself, and pathways responsible for the growth and maintenance of blood vessels, including the extracellular matrix. So this overall downregulation of these proteins does suggest that even in people who carry the CADASIL-related Notch3 variant but have very mild or no symptoms, there are already signs of altered molecular activity. And it points to disruptions specifically in immune and vascular processes that could be playing an early role in disease development. Notably, we didn't find any proteins that were able to predict the occurrence of stroke or dementia, but still, I think it's important to point out that the fact that these protein changes appear before any clinical symptoms does suggest that there is a window in which intervention may be possible, even before structural brain damage or clinical events occur. And if we take a closer look at one of these 11 proteins, for example, TIMP3, we can see how many different processes it's involved in, like inflammation, angiogenesis, extracellular matrix remodeling, among others. So again, it's important to ask, how is looking at its behavior in Notch3 variant carriers helpful? Well, we know that Notch3 mutation, we know that the Notch3 mutation causes abnormal buildup of proteins around small blood vessels in the brain, which we can see from sort of my initial analysis here seems to interfere with the production or stability of other important proteins like TIMP3. Now, TIMP3 is a protein that helps blood vessels stay healthy by regulating inflammation and supporting normal blood vessel growth. So when we have TIMP3 levels that are lower, blood vessels could become more fragile, more inflamed, and less able to repair themselves, all of which could contribute to the early stages of damage we see in CADASIL. So by looking at protein levels in variant carriers compared to controls, this can point to which specific pathways are affected and reveal more about what is driving the disease on a molecular level. And this information is valuable in a variety of ways. For example, if we know that certain proteins involved in blood vessel health are affected early in CADASIL, we can look at existing drugs that target these proteins or pathways to see if they could be repurposed to help prevent or delay the disease. Or it could point to new targets for treatment that could be developed in the future. So now I'll briefly go over what we found when we looked at metabolite levels. Overall, it seemed that protein levels differed the most between variant carriers and controls compared to metabolite levels, but there is an interesting trend that I wanted to point out in terms of the metabolic markers that we looked at. So overall, we found 26 metabolic markers that differed between people with the Notch3 variant and those without. And of these, 19 were associated with the overall occurrence of migraine, stroke, or dementia. However, like the proteins, none of these were predictive associations, meaning they weren't strong enough to reliably forecast whether someone would go on to develop these conditions. Now, what I found interesting was that most of these significant metabolic markers involved lower levels of a particle that is essentially high-density lipoprotein, or HDL for short. So HDL particles are essentially good fat molecules that help clear cholesterol from the bloodstream, indicated by these little blue circles here. So these lower levels of HDL might suggest that people with the Notch3 variant have a reduced ability to clear cholesterol, which could lead to buildup and potentially damage small blood vessels over time. But I also want to point out that this preliminary analysis only included about half of all Notch3 variant carriers in the UK Biobank study, because many participants didn't have blood data available yet. So this data is expected to be released quite soon, and once it is, I'll be able to repeat the analysis using the full data set of just over 1,000 Notch3 variant carriers, which should hopefully give us a clearer picture. So, so far, my research this year has focused on analyzing data from the general population, which typically includes much milder cases. However, it's really important to investigate these molecular profiles, particularly proteins that showed promising results, like TIMP3, in a patient cohort with individuals who have confirmed familial small vessel disease, where cases tend to be more severe. So I'll soon begin analyzing blood samples from participants in our UK familial small vessel disease study, which is based at the Cambridge Clinic. And right now we have about 260 blood samples from CADASIL patients that are ready to be processed. However, we're still in the final stages of securing funding to cover the processing costs. So far we have about 80% of the total amount needed, with about £24,000 coming from our Cambridge CADASIL Fund, which includes donations made over the past several years, £16,000 from my PhD studentship, and about £15,000 that was raised through CADASIL Support UK through a special call for research funding that they very generously led. And so we're now in the process of seeking that remaining 20%, which is around £16,000 to proceed with the analysis of these samples. So I think it's important to stress that we're really immensely grateful for everyone who has generously donated. This support really plays a crucial role in our ability to carry out this research, which we hope will make a real and meaningful impact on patients' lives. So my overall aim with this research, just to reiterate, is to identify predictive tools for CADASIL. And so to achieve this, it's essential to compare molecular data from blood samples with established markers of clinical severity, particularly objective quantifiable markers measured at multiple time points that reflect the severity of the disease and how it's progressing. And imaging features from MRI scans, such as lacunar infarcts and white matter hyperintensities, which are the imaging features typically seen in CADASIL, are ideal for this purpose. So this longitudinal approach allows us to assess whether an initial blood sample can help predict how the disease will progress in an individual patient over time. So because of this, one of the other main projects I'll be doing is a retrospective longitudinal MRI study to better understand how CADASIL progresses and to identify possible early predictive tools. So this means I'll look back at changes in key brain imaging features over time in patients who have had multiple scans. So as I mentioned earlier, most of our CADASIL patients have at least one MRI scan, and many have had multiple scans over the years. So I'll be looking at changes in things like the number of lacunes, white matter changes, microbleeds, and changes in brain volume, and seeing how these relate to clinical outcomes, such as future strokes, dementia, cognitive decline, and levels of disability, as well as, of course, exploring how these brain changes really relate to molecules in the blood. So these projects all contribute to my overarching goal, which is to develop a risk model that combines blood data, brain imaging, and clinical information to generate personalized risk scores. So these scores would help predict how severe the disease might become and how it may progress over time. So the aim is really to improve prediction beyond traditional factors like age, mutation type, and known cardiovascular risk factors like smoking. So this will not only help us understand more about what is driving CADASIL, but it will also improve patient care by making it more personalized. It'll help us identify new treatment targets, and it'll improve our ability to test these treatments in an effective and reliable way. So I'll be comparing this new model to the few existing ones, and ultimately my goal is to turn this into a tool that can be used in clinical practice. And this last part, making sure the tool is understandable and useful in the real world, is where the involvement of patients and families becomes especially important. So that includes all of you here today. So I hope to work with anyone who's interested in being part of something like a patient advisory group. For example, I'd love your input on how best to present the results from the risk model once it's developed, whether that's through something like color-coded risk categories, timelines, or something else entirely. That's just one of the topics that I'd want to discuss. So I'm currently collaborating with CADASIL Support UK about how best to set this up. But there'll also be a financial reimbursement for anyone who takes part in these discussions for their time, which is supported by the Stroke Association as part of my scholarship. So I really look forward to hearing your thoughts on how this research can have the most meaningful impact for CADASIL patients. So with that, thank you so much for listening, and I really look forward to any further discussion. So thank you very much.
Thank you so much, Natalia. That is really, really interesting. Lots to take in for people. But it's nice to see that you're doing so much, and it's really nice to see that Stroke Association has felt it's very important because they interviewed you for it, so it was quite a you know, comprehensive panel and assessment and things. So it's great to see that. So you got some clapping hands. But if anybody would like to ask you any questions, that's absolutely, you know, fine, or we can ask questions later. And you know, if there's anything people would like to ask, if not, we can just talk about things a bit more, and you can think about questions as we go along. Just to remind you that the chat is on the bottom, So you just click on there and you type in if you want to message anything there. So maybe I think probably what we'll do is just talk a little bit then generally about things. But as I say, do ask as we go along. So what I'm going to do is just, so one of the things people asked was whether we could tell them a little bit about, what some of the funds that people have raised have gone to. So you've heard one great project from Natalia. So, but what I'm going to talk to you is a little bit more about some of the work we've done in the past. And I think that what I would like to emphasize that is even more important than money raised is that people can actually take part in the research, because without that, we just couldn't get anywhere. And I think you'll see that when I present some of the research. So what we try and do in the group is we do two things. One is we try and tackle common clinical problems and see whether we can better manage them. And then we also try and look in much more detail about what causes CADASIL and how we treat it. Now, the first half we're going to get answers much quicker from. The second half will take much longer, of course. First of all, just to say that we did get quite a lot of press coverage of one paper that we published recently. So I got in BBC News and many other things. And some of you may have seen Glenn in various forms, either in sports kit or with his cars and various things, which is really helpful. So I think the more we can do to promote CADASIL in the public arena, the better, because it gets, when people realize what it is, we're likely to get more support for it, both from the health service and also from people raising money for it. So this was actually the project that we that got all the attention, because it had a press release from Cambridge, and this is what got the BBC and other people interested in it, which is really good news. So this is this study that Pat, many of you may remember, did some time ago, and it now came out in its final paper, and just shows that the outlook for people with CADASIL has improved a lot over the years. So this line shows you the sort of most common age of onset of strokes in people with CADASIL, and you can see that in the sort of 30-year period, it's gone up by about 15 years. So the age where people have their first stroke is now about 15 years older. Now, this is due to a number of things. One is obviously that, we're getting milder people, but it's also due to improved treatment of risk factors such as high blood pressure and smoking and so on. The second thing, just to point to this, is actually each of these dots is one person. So there's a big spread of severity, as we've discussed before. This is Pat's paper, which is freely available, so anybody can get it. I'll just leave that up there for a moment if you want to download it from the code. Here he is presenting his poster, his presentation to the Princess of Thailand, because he got a Royal Scholarship to come over. So this is sort of the key summary. So a lot of the papers now, when you look at them, have this sort of key point summary, which is partly designed for people who aren't so aren't from the academic field but want to know about it. So I think this tells you, stroke used to occur in the 40s. Now the risk has increased both for stroke and dementia. The age has increased for stroke and dementia. And also, as I mentioned, if you read the paper in more detail, you'll see that it really does emphasize the importance of treating cardiovascular risk factors in CADASIL. So this research has really come from this CADASIL research database. So people often ask in clinic, how can they contribute to research? And actually, by joining the register, this means that we carry on using the data for many, many years. So even though it may only take you an hour, many of this has gone into sort of 5 or 10 projects and is still data being used. So many of you have taken part in this already. So it basically involves a questionnaire, brief memory, cognitive testing, mood questionnaire, a blood sample. It takes about 45 minutes to an hour. And then it allows us to get all of the data which you've got on your records, so your scans, your clinical data, GP follow-up, etc., hospital records, to really get a lot of data out over time. And we've now got over 600 people who have joined the register, and it is the largest CADASIL database worldwide. So I've tried to put on here a few of the things that the database has told us over the last years. So perhaps one of the most important was that things like smoking and high blood pressure accelerate the disease. This wasn't thought to be the case. It was thought that CADASIL primarily affected people who didn't have risk factors. That was wrong. And it's been shown that these risk factors accelerate the disease. And it's also increasingly been shown that if you treat these risk factors, particularly early on, the earlier the better, it slows the disease. Doesn't stop it, but it slows it. We've done quite a lot of data on looking at how the site of the mutation relates to the disease severity. So many of you have come across this, is this EGFR, which EGFR in the gene the mutation's in. And Amy has put a calculator on. So if you want to calculate this, you can go to our website and find it out. Amy did quite a lot of work on fatigue, showing how important it was, how common it was, and what the sort of factors associated with it were in CADASIL. And this has now led us to us doing a clinical trial of a new treatment for fatigue called modafinil. We're doing it actually in all strokes because fatigue is a problem in, but not only CADASIL, but in stroke generally. So hopefully we'll know whether this drug will help. This is a study in a few hundred people that's led from Australia. More recently, we looked at brain hemorrhage in CADASIL, which can occur. Not commonly, but can occur. So we know much more about this. Current projects we're currently looking at infection. And what we've noticed is that some people, including quite young people, can get multiple little strokes. You can see the brain scan here in the little white spots. They're tiny little strokes that with infection, particularly with COVID. So you can, and they can sometimes recover fully, but sometimes can lead to significant impairment. So this is really emphasizes the importance of vaccination. So COVID-19 and influenza, which I'd strongly recommend you ask for. We've mentioned before that we're doing this study to try and see whether we can identify other genes that affect the severity of CADASIL. But I'll just recap this because this is one of our big studies at the moment. So this follows on the fact that, as I showed you from that, all those little dots before, the severity of CADASIL, even within families, is really quite variable. And if we could identify what causes this variability, it could improve treatment. And one of them is things like hypertension and smoking. But we also think that there are other genes that everybody's got, and this interacts with the CADASIL mutation. So what we're doing here is we take out DNA and we look at the markers of the DNA over the whole of the genome. So we can look in lots of different genes, and then we can identify the genetic makeup in different people. So what we then are trying to do is we're doing this in lots of people with CADASIL and saying, can we identify any other genes that affect the severity of CADASIL? So what we do here is we collect as many cases as we can. And this is exciting because it's, one of the first really large projects worldwide in CADASIL, which is what we have to do if we're going to improve care. And then we try and look at genes that are related to determine when you get your first stroke in CADASIL, or if you've not had a stroke, what protects you. And we also look at the amount of damage to the white matter, the deep structures in the brain. And you can see here the red. We have sort of software that counts the amount or quantifies the amount of damage. This is something that Amy's helping with and coordinating, and this depends on us getting everybody together. So we're really fortunate. We've got really enthusiastic people worldwide, good people. We had a meeting in Cambridge. This was a meeting in Paris, and the next meeting is in Leiden in the Netherlands in June next year. And this has allowed us to get almost 3,500 cases of CADASIL. 600 of these are from our own database, but as you can see, a lot more. And we've now got people with MRI scans in about 2,500. So it's a really big database. So it gives us a lot of power to answer these questions. So just a little bit more, what else do we need to know? What are we really wanting to know? I think there are two main things. One is how we can treat common symptoms better. So we've shown that fatigue is common. We've done quite a lot of work on apathy, which is this lot of loss of motivation, which can be, quite disabling, particularly for partners and other family members. And then we want to get new treatments. So just a word on new treatments. So, what happens in CADASIL is you've got your abnormality in your DNA, one little tiny abnormality, which then leads to an abnormality in the coding of proteins. And proteins are your building blocks in one's bodies. And so ideally what we would like to do is actually correct the DNA. This is quite a challenge, but increasingly people are beginning to do this in diseases. So you can either sort of try and repair the DNA itself, or you can actually just use some way of reducing production of that protein if it's causing problems. So what people are now doing is that there are some very exciting techniques where you can actually correct the DNA itself. Now, this is a little way off in CADASIL, but it's starting to encroach on a variety of diseases, particularly at the moment, blood diseases, because you can take out all the blood cells, you can correct them and then inject them back into people. It's harder with blood vessels because we've got to find a way of actually correcting the blood vessels themselves. But it is certainly the future, and people are trying to do this in CADASIL models in a dish to see whether they can correct the CADASIL abnormality and then improve function. So that's an exciting area. The second thing you can do is you can try and reduce the amount of protein that's produced. So you can sort of block it at this stage, what we call the messenger RNA stage, and make less protein. And you can use these things called ASOs to block it. And these block this production of the proteins from messenger RNA. And these are increasingly being used in neurological disease. I think I mentioned this year in our in-person meeting. So they're being used in a disease called also neuromuscular disease called SMA, muscular atrophy and spinal muscular atrophy. And they've been successful. They're now licensed by the NHS. So what we are hoping is that people, that we could get companies interested in applying this to CADASIL. There are, of course, challenges in these therapies. So, big challenge is you've got to make sure you edit the right cells. So getting these techniques into the right cells in brain blood vessels are tricky. So people explore things like giving sort of viruses that don't cause problems that will then transfer it into the cells. If you give something like these ASOs, you have to inject them into the spinal fluid. And then what we need to do is we want to make sure we don't sort of cause other effects that we don't want. So it's all about balancing risks against benefits, but it's certainly a very exciting area. And the other area that's really being taken forward at the moment is another area I did mention before, which is so what happens in CADASIL is you get these aggregates that form in the blood vessels. And this then disrupts the blood vessel function, as Natalia very nicely showed. So, and what you can do is you can clear these aggregates by making the body react against them by vaccinating it. So you actually vaccinate against these Notch3 deposits. And then this helps clear the abnormal protein. And we did discuss this at the meeting, but it might be that, if people are interested, we could get somebody to talk about this at one of these meetings, perhaps, and who's doing the research and explain how it's working. Alternative approaches, there is interest in repurposing drugs we already have or developing new drugs, and people are trying this at the moment. So maybe we'll have some success here. So that gives you just a little insight into where things are going. But just to emphasize again that, the research really wouldn't be possible without people agreeing to take part. Absolutely, that's the only way we can do it. So thank you very much for that. If you'd like to be involved, we do have this register of CADASIL patients. It also gives us permission to contact you later if there are other projects we want to contact you, because we do need permission to keep your name on the sort of recall database. Just to mention, we do need you registered as a patient at Addenbrooke's or in another hospital involved in the research. So we can't really do research on people from abroad, for example, just because of ethics reasons, but we can put you in touch with people in other countries who could then include you in like the big project we're doing. This is just telling you, what we've spent money on. So, you raised about 25,000, I think it was, for some, which helped Alex Granata a lot with the CADASIL in the dish. I'm not going to talk about this because Alex presented it and there's a very nice video on our website. We've talked about the proteomic study, and then we do use some of the money on things like the CADASIL information leaflets and website. We used to use it on the annual meeting, but we've actually managed to fund that from other sources recently because people are keen on patient and public involvement. So we've managed to fund that separately. So we haven't had to spend money on it recently. If you do want to take part in research, then do contact us or contact CADASIL Support UK. Just to say also, we've also, the fund, the pilot research we do in the fundraising does help us raise other research. You've heard about Natalia's studentship, but we've also recently got about over 300,000, I think it's about 350,000 from Alzheimer's Research UK to carry on with our CADASIL in a dish work. So it really does help us get more money, because in the end, this is the sort of money we need to do the projects. So just moving on now to one of the questions. So this is, we'll have questions after the break, but this was one of the questions. And I think it would be quite helpful just to show you how we're doing the fundraising at the moment. Now, we used to raise money via an account called the Addenbrooke's Charitable Trust, because what we want to do is always raise money via a charity, partly because, it's transparent and the appropriate way to do it. And the second thing is that it allows people to get tax relief on it, which gives more to the donations and also can reduce people's tax bill. So, and we used to do this via CADASIL Support UK and also via Addenbrooke's Charitable Trust. That became a little complex, I think it's fair to say. And also they did take some of the money to support the runnings of the charity, which over time could add up to, 10, 20%. So we've now organised a new way of doing it, which is through the University of Cambridge charitable funding, which again allows you to give money via links and with tax relief, but it does mean that 100% of the money can be spent on CADASIL research, and also it's easier to manage. So I think, Amy, have you prepared something on that? Or do you want me to talk about it?
Yep, I have a short presentation, a walkthrough.
I think probably what I'll do is I'll pass it over to Amy, because I think I saw she prepared something on it. So it'd probably be better. So let me do that, Amy, for you. And maybe you can just take that over.
Yeah, no problem. So, as Hugh has said, we have recently moved our donations from ACT Charitable Trust to the University of Cambridge. So yeah, all donations should now be made via the University of Cambridge. We have updated all the information on our website on how to do this, but I will just quickly walk you through how to do this on our website just to make it really clear. It's a very straightforward process. So if you go onto our website, cadasil.co.uk, you can see at the side we have, I'm not sure if you can see my mouse.
Yes.
Brilliant. So you can see we have lots of different categories of things here, and there is a section called Donate to CADASIL Research. So if you click here, this will take you to our donations page. You can find some information on online donations and check donations here. There's also a link at the bottom to look at some of our past supporters, although this is a bit old, it should be updated. So for online donations, this is probably the easiest way to donate. So to do this, you would just click here, donate here, and this links you straight to the university website, which looks like this. Now, this is a general giving page for the university, so you will have to specify that your donation is for the CADASIL Research Fund, and I'll show you how to do this coming up. But you can see on this page that you have the option to select whether your donation is a one-off or whether it's recurring, and then you can enter the gift amount here. You'll then be prompted to enter your personal information. And at the bottom of the page, there is a box that says what has prompted your gift today. And in here is where you should specify that the donation that you're making is for the CADASIL Fund. And you should just put in here Prof. Hugh Markus Research, CADASIL Fund, RREX, EFKM. And this is on the donations page on the website as well. So you can just copy it from our page onto the box here. And this just makes sure that your donation comes to us rather than the department in general. And also just to say that if you're eligible, please do tick the gift aid box, as it increases the amount that comes to our research fund. And check, so you can still donate to check. Please specify this same fund, so also specify Prof. Hugh Markus Research, CADASIL Fund, RREX, EFKM, and then you can send it to this address here, CADASIL Fund, Department of Clinical Neurosciences, and this is just our postal address. And again, this information is available on our website. And hopefully that is all straightforward, but if you ever do have any questions, you can always email me and I'll be very happy to help.
Thanks, Amy. So just to emphasize a few things, you can also give money to CADASIL Support UK. So they've got a good system for that as well. If you do give it to Cambridge, make absolutely sure you put that information Amy asked you to put in, which account it goes to, because we don't want it just to go to the university. So it's very important. And I think it's much easier doing it online than checks, because checks can always get lost and things, and if you can do that. But I think that's great.